So I want to start off by saying thank you to Ann and Greg for inviting me to contribute to the symposium. It's a great honor. Thanks to all the DLC staff for organizing this. And all the speakers today just did a fantastic job, and you're really a tough act to follow. It's tough to be the last speaker of the day. I'll do my best. I'm starting off with this globe. I'm keeping in the theme of the One World, One Health approach, right, that both Meredith and Ph.D. talked about. And the angle, though, that we're taking with our project is a little bit different, and we're using next-generation sequencing to discover pathogens that may be circulating on Madagascar that people never even knew existed before. And the One World, One Health approach really brings together a team of people. And so everything I'm talking about today is really a team approach, and everyone that I'm listing there has played a very important role in everything that I'll be presenting. Meredith's talk just took out the first slide of this presentation, so she did a great job. What I'm trying to show here is that there are a number of really negative pressures that are impacting lemur health and lemur diversity on Madagascar. The map is showing habitat suitability, and this is from a paper from Jason and Ann last year. And the warm colors are showing areas that are highly suitable for a number of lemur species. And all the things that Meredith talked about, climate change, slash and burn agriculture, those are serving to disrupt lemur populations. Populations are getting more dense. We have to think about parasites. We have to think about pathogens within lemurs. And so it's really a dynamic situation for lots of reasons, and it's depressing. About a year ago, I woke up one morning and I told my wife, I was like, I just had the coolest idea for a figure, and she said, you need to take a break, right? So I rushed into the lab, and I got Ryan Campbell, a graduate student of Ann's, and we drew this out on a whiteboard. The idea for the figure was it would be so interesting to visualize the IUCN status of every species of lemur all at once, all in one image. What kind of patterns would we see within that figure? And so across the top, I'm showing the IUCN status in different colors there. The blue is least concern, green, you're threatened, vulnerable, endangered, critical, right? And along the bottom, I'm showing about 100 species of lemurs. This figure is a few months old. There's new species that have been described since then, but those are arranged by taxonomy, by family. And so if we just work through this, right, there are 3 least concerned species, and so I'm going to show that we have a source and a target. And so we'll go through this figure step by step. You can see near threatened, and then we're going to get into vulnerable, and then this is really amazing, endangered, right? And so that stat of 94%, right, endangered or critically endangered, we're going to visualize all in this at the same time this figure, data deficient. And the data deficient ones are scary because we don't really know anything about those species, right? So this is what it looks like all at the same time, right? So there's a lot of red there, a lot of warm colors, and that's really showing the situation that we are in with respect to lemur health, right, and lemur conservation. But there's hope, right? And so I like to do this, and there's hope, right? There has been amazing revolution within the last few years with respect to genomic technologies, right? And so this is just a great title that Ann had, and this is really a fun time to be a biologist, to be an evolutionary biologist, because it's the Wild West of genomics. Right, next generation sequencing technologies, new technologies are coming out all the time. This is an area of rapid development. And so the question is, how can we use all these really fascinating technologies, right? So my go-to line here is, how can we leverage the power of these tools to accelerate pathogen discovery in Madagascar? How can we use these tools to inform a One World, One Health framework, right? And then how can we do that also to learn more about lemur conservation efforts? Right, so again, we can use these technologies in new ways for lemur conservation. We can use these emerging genomic tools to aid lemur conservation strategies, right? And so this is the framework of my talk today. When we look at Madagascar, think about, and both Meredith and Ph.D. mentioned this, think about diversity on Madagascar, right? We know so much about the faunal diversity, but also think about the pathogen diversity. What has evolved in isolation on Madagascar over the last 135 million years, right? What pathogens have arrived to Madagascar via human colonization over the last couple thousand years? What pathogens arrived with the animals that humans bring with them? The cattle, domesticated species, cats, dogs, the ticks on those animals, right? How about the rodents that jump off the ship at the port, right? The fleas that they carry, and that's why plague is an issue. And so really, do we have a good understanding of pathogen diversity on Madagascar? And which diagnostic tools do we have to look at that diversity? There's a failure rate with PCR. There's a failure rate with ELISAs. And so what methods exist out there to really get a greater understanding of what is going on in Madagascar? And that brings in next generation pathogen discovery, right? So it's using these emerging tools to deep sequence DNA and RNA from biological sample. And what's so interesting about this approach is that you don't need any information up front. You don't need any PCR primers, no probes, nothing. All you need is a good biological sample. And with respect to lemurs, you can do this type of research in a minimally invasive way. All you need is a blood sample, right, or even a fecal sample. So once you have that sample and you deep sequence it, you can then bioinformatically identify all the bacteria, the viruses, the fungal sequences in that sample. And that's a metagenomic approach, like what Aaron mentioned. So from the One World, One Health and conservation perspective, detection is the first step, right? We can identify that a parasite is there, but whether or not it causes disease, that requires further research. We can use this type of approach in a phylogenetic framework as well, right? So we can use phylogenetics to determine if a species or a strain is closely related to known disease-causing agents, and that tells us something. We can use that information then to design rapid diagnostics that are cheap and can go through hundreds of samples, right? And so this is what we are establishing in Eastern Madagascar. It's a One Health project that's really rooted in next-generation discovery. And there's several phases to this project. We've only reached phase 2, but phase 1 was to use next-generation sequencing to discover parasites that may play a role in disease. And we did this with a pilot study that started very small, but is turning very big. The second phase is to develop a cost-effective assay to rapidly screen hundreds of samples. We've done that, and we just got the data within the last few days, and I'll highlight some of that. But the third phase is then to work with the One Health team to really determine pathogenicity, vectors, reservoirs, really put this together in a greater framework to understand what's going on the ground in Madagascar that may be negatively influencing human health, wildlife, and agricultural species. And throughout this process, we really need to inform lemur conservation efforts to local clinicians in Madagascar and the greater One Health community of our discoveries. So it was about 2 years ago where I was at the Lemur Center. I saw Kathy give a talk, and she was talking about all the work that had been doing monitoring lemur health in Batavia, that red dot there in the eastern part of Madagascar. And one of the things that she said is that they weren't really identifying pathogens in the blood samples, that the assays that they were running were consistently negative. And my background as a mammologist, and I've done some disease surveillance work in the field, I knew that there were probably things in those samples that just weren't being detected, right? Their methods were probably biased. And so I talked with Kathy and I said, let's develop a pilot study where let's just take 6 samples, 3 injury, 3 diadema. Let's take 6 samples and let's deep sequence those and see what we find. And we just wanted to make sure that we could even, that the project was feasible and just to get it started, right? So this is what we did. Kathy acquired 6 blood samples. Again, very small pilot study. You can see that on the map there, the blue dots are from injury, the red are for Propithecus, and these are sampled around an active nickel mining site at Batavia. And there's lots of human pressures on the environment surrounding this mine. Right, and so we really didn't know what to expect with the level of habitat destruction around this area with the kinds of parasites or pathogens that we identify in our sample. So it was very straightforward. We chose blood transcriptomics for this approach. We deep sequenced RNA, and there's several reasons why we did that. But we extracted total RNA from those samples, then barcoded and pooled those into a single library and deep sequenced those on one Illumina high-seq lane, right? So this is like the bare minimum, 6 individuals, one lane, one library, one lane. The sequencing resulted in 37 million read pairs per individual. That's pretty much standard. And now with the latest developments, it gets much greater throughput than that. But we developed this bioinformatic pipeline for de novo parasite identification, and this is highlighted in the paper that was published this year in Biology Letters. But basically what we can do now when we generate sequence data like this is that we can go from raw data to a putative taxonomic identification in less than 24 hours, right? So the pilot study really helped us put together a pipeline that could be used in the future. So what did we find? Well, we found lots of things, if they would come up here. 3 new species of Plasmodium. We found a new species of trypanosome. We found multiple tick-borne parasites, some of which were never even known to occur in Madagascar. And remember, this is 6 individuals, right? We know that we're highly confident that we identified unrecognized species diversity in Plasmodium and trypanosomes because there's a great body of work that has been done on Plasmodium in Madagascar especially, right? So we were able to take our sequence data and put it into that body of work and know that we had identified new lineages of Plasmodium. And the same is true for the trypanosomes. But what was really cool were these tick-borne pathogens, parasites. And that is, it's because some of these, like Neoerlichia, were not even known to occur in Madagascar before. And so we took a really close look at those, and that's what our first paper really addressed, was the variation of these tick-borne parasites. And again, if we just look at those 6 individuals, and we look at those, how those parasites, the infection rates of those parasites across those 6 individuals, it's a very small sample size. We could see that Babesia was common, Plasmodium was very common, and that pattern has been identified recently in a paper last year, this common prevalence of Babesia and Plasmodium in wild lemurs. But what it resulted in, what we achieved was we sequenced entire ribosomal and mitochondrial genes, and that was really important because that allowed us to put our sequence data into a phylogenetic data set. Much of the work on tick-borne pathogens, they use ribosomal sequences for phylogenetic analyses, and so it was really nice to take our de novo approach and then put it into those data sets to look at genetic variation. And I'll show you the phylogenies here in a little bit. But it seems like the patterns that we're recovering is that the tick-borne parasites were really closely related to those that are known to cause disease in humans, livestock, and wildlife, right? And so the samples were sent to our collaborators at North Carolina State Vector-Borne Disease Lab for independent confirmation of what we had identified from our next-gen approach, and they did that using DNA. So we're going to take a closer look at these tick-borne parasites. We don't know if these are causing illness in lemurs now. What we can tell you is that some of these are very closely related to strains that do cause illness. And for Borrelia, this is a bacterial spirochete. It's a causative agent. There's a species of Borrelia that's a causative agent of Lyme disease. But this causes borreliosis in wildlife and livestock, and this is the first sequence that we know of from a lemur. The Babesia, this is a malaria-like preoplasm, and Babesia causes babesiosis. This is symptoms similar to Lyme disease in humans. But this is an emerging disease, not only in humans, but in wildlife, emerging disease in cattle. There's several strains of Babesia that have been identified in dogs, right? And it really seems like Babesia is fairly common across lemurs in eastern Madagascar. And this has been noted by our study and then also a study that was published last year by Springer et al. But what was so surprising with this Neoerlichia, Neoerlichiosis is prevalent in humans, but this is the first record for Madagascar. And Neoerlichia is something that's only been known for a couple decades now, and there aren't very many sequences that have been generated worldwide. So we really don't know, as a health community, we don't really know what Neuralichia, how that's impacting wildlife, agricultural species, and even humans. So if we look at these phylogenies, at the top there is Babesia, and the Babesia in lemurs is appearing right here. And what's really cool in light of Ph.D.'s talk is that this sequence is nestled right among sequences that were of Babesia that were gathered from domestic cats in South Africa, right, and lions in Africa, as well as a couple of baboons that were wild caught, right? So it seems like there's this connection here with domestic cats with respect to this Babesia species. For the Borrelia, that sequence clustered with sequences that were gathered from ticks that were collected from cattle in Mali, and also, I think, from a cow in Japan. So it seems like there's this connection with cattle. Right, and so this is indicating potential spillover from domesticated species. And finally, the Neuralichia. So again, almost nothing is known about Neuralichia. You can see just a couple of sequences on GenBank at the time of the study. And actually, about 2 weeks ago, I was emailed by a researcher in Australia who said, Peter, I just sequenced Neuralichia in Australia, and it is sister to the species that you have in Madagascar. So it seems like there's this really interesting connection with Neuralichia and Australia fauna, right? So there's so much work that needs to be done on that. So phase 2, we used those sequencing results, right, of the pilot study to design quantitative PCR probes, right? And so that allowed us to screen, well, we've screened about 95 samples at this point. But if we zoom in and look at this study site, right, so Kathy and her team over the last few years have collected over 120 samples, right? So we can use those results from the NGS approach to screen all of these for those tick-borne parasites we identified, right? And so we can go through and look at then the results of those qPCR assays. And again, for some of these, so we expanded to 4 species. There's Avahi, injuries there, Lemur. But look at Propithecus diadema, 69 individuals, right? 84% of those were infected with Babesia. And look at Neuralichia in Propithecus diadema, 29% infection rate. That's pretty remarkable considering that Neuralichia wasn't even known to occur in Madagascar before we identified it in our sequence data. And there's so much that we can do with this information. Co-infections, there's an injury and 5 diadema individuals that were infected with all 3 parasites, right? So we just scratched the surface here. We just got these data in a few days ago. And if we look at how these are distributed geographically, you know, Babesia is very common. These are 58 individuals of diadema. They're in blue around the study site. Injuries in green, Avahi's in turquoise there. And what's really interesting is that the Shafak shows this heavy tick burden in the nostrils, right, by Haemaphysalis lemuris. And this is something that Klopman et al. Noticed, I think, a couple of years ago. What we're seeing, it seems like the data that we're generating is that the Propithecus, the Shafaks, have this very high prevalence of tick-borne pathogens associated with them. And so we don't know what the tick vector is yet, right? But what we can do now is we can take the ticks from these nostrils and we can screen those, right? And we can identify whether or not that species is playing a role in this transmission. The Borrelia, weren't really many patterns that really just came out. It's too early to say across the geographic area, but we can just see how those are broken out geographically. And then finally, the Neuralichia. So really, and to build off of what Meredith presented on, we don't really have a good understanding about emerging tick-borne pathogens in Madagascar, right? In just one pilot study, just 6 individuals, we uncovered this incredible diversity, right? And with all the pressures that are going on, we really need, as a One Health team, we really need to figure out more about whether or not these pathogens may be causing disease in humans. If you think about bushmeat, people coming into contact with wild lemurs, right? Spillover from agricultural species. There's so much work that needs to be done, and all this is very dynamic because of climate change, right? So it's really just an area of research that must be looked into. Well, we can go back now and we can use information, right? We can use like the ticks of Madagascar, for example, to look at which ticks are distributed or occur there, how are they distributed, and we can target those with these sequencing approaches to identify pathogens that may be circulating in them. Again, we don't know if they cause disease yet, but there are records of borreliosis and babesiosis in humans and in livestock in Madagascar, but they don't know what's causing that, right? So it could be that something that we identified is causing disease in Madagascar, right? Which tick species are serving as the vectors? What is the prevalence in lemurs? How are these pathogens distributed across the island? And really, Madagascar clinicians and vets, they must be aware of these novel tick-borne parasites, because if people if patients are displaying symptoms that are consistent with a tick-borne disease, right, it could be that something that we've identified is playing a role in that. So we use next-generation sequencing to discover multiple parasites that were previously unknown to be circulating in lemurs. And the same approach can be used for virus discovery. That's one direction that we haven't gone yet. We decided to focus on the tick-borne pathogens, but the same methods can be used for viral discovery. And what's really exciting is that future conservation teams can use similar methods with advanced portable sequencing technology like the Oxford Nanopore MinION for rapid real-time diagnostics in the field. People are already doing this in Africa with Ebola research. And I mean, we're approaching a tricorder essentially, right, from Star Trek, because we can use this sequencing technology in the field. You don't really need very advanced equipment. You can use a high-tech discovery approach, followed up by low-tech, low-cost screening across many samples, right? So we are going to travel to Mbatvi in March of next year. We're going to collect tick and blood samples from cattle, dogs, lemurs, and we are going to identify the tick vectors involved with these pathogens. We will build on the next-gen approach, all right, and identify whether or not there are other tick-borne pathogens present at our study site. And we're going to collaborate with ecologists and local veterinarians to really get a good understanding of what's going on. And as I mentioned before, we really need to share these results with clinicians in Madagascar and with the One Health community to really understand whether or not these are causing disease. All right, there are so many people to thank. Of course, I have to thank Anne and all her support and everyone in the Yoder Lab. The original 6 individuals, to thank them. Funding from TriSim, this really couldn't have happened without a grant from TriSim that allowed us to screen these initial 95 samples. And just thank you so much for your time.